Hello everyone, I'm Alejandro Montenegro. I'm an executive editor for Cold Spring Harbor Protocols. I'm joined today by Maritxell Huch, who is the director and group leader at the Max-Planck Institute of Molecular Cell Biology and Genetics, to talk a little bit about their research and the work they will present at the 87th Cold Spring Harbor Symposium, which this year focuses on stem cells. Thank you very much for being here.
Thanks for the invitation.
So work in your lab is centered around adult tissue homeostasis and repair and their deregulation in disease. And one of the strategies you use is the development of a very specialized type of 3D model to study tissues and organs on a dish. I was wondering if you could tell us a little bit about that.
So indeed, since I was a postdoc, we developed these 3D organoid models directly from the tissue, not from pluripotent stem cells. And we realized when I started my lab, which before was in Cambridge, now I moved to the Max-Planck, we realized that we can use them to understand questions and principles of basic tissue regeneration, and how when these principles are wrongly regulated and how they become into disease.
Mm-hmm. So what advantages would you say these kind of systems have versus normal 2D culture systems?
So of course organoids cannot be used for everything, but they have the advantage that they retain the normal physiology of the tissue in the dish, many aspects of it, not everything, but many aspects of it, with the advantage that there is no genetic mutations. These are not cancer cell lines. This is primary tissue not transformed that we take from, let's take the example of the liver. You take a piece of the liver, you put it in culture, and this tissue that you had in the organ, in the animal, or in the body of the human is retaining these same properties in vitro in the dish, meaning that they do not have genetic aberrations, but we can maintain them long term, which allows us to now perform many experiments from when we use patient-derived organoids, meaning that from a patient that, for instance, has a cancer and we develop cancer organoids from this patient, now we can use them to test drugs, or we can also use them to investigate gene expression or the impact of a particular gene in a regenerative program, for instance.
Mm-hmm. So you center your talk around the liver and how the system is amenable for studying adult tissue regeneration and disease, and that this could be done at different scales. I was wondering if you could tell us a little bit about what you meant by different scales.
Yeah, so the way we think about regeneration is an holistic process. So basically the cells have to sense that there has been a damage and they have to repair, but then this repair means that inside of the cell, intrinsically, the cell has to change its molecular profiling to activate the regenerative program. So this is the molecular scale. But at the same time, it has to repair the tissue, it not only has to proliferate, but it has to also establish new contacts with the cells and reestablish the contacts that has been lost. This is the cellular scale. And we also now start to realize that actually there is also tissue scale properties that are important and they needs to be repaired during the damage repair response, like recruiting cells from other parts of the tissue, for instance. Mm-hmm. So this is what we mean by the problem of regeneration is a problem of scales.
Mm-hmm. In your abstract, you mentioned that you have been able to expand these organoid tissue cultures for months. Yes. Defying what is known as the Hayflick limit. So I was wondering if you could tell us a little bit about what that limit is and what the implications are of defying it.
Yeah, so the Hayflick limit was established in the 60s by Hayflick himself, who realized that if you want to expand primary cells, not transform, not cancer cells, but primary cells in a dish, basically was not possible. And every time he managed to expand them, they were fibroblast type of cells. But epithelial cells were not possible, unless they were transformed, they were coming from a cancer. So it was our surprise to realize that, and starting from stem cells in the gut and stem cells in the stomach, we realized that we can isolate these stem cells, and actually in our body they retain this capacity to divide for our entire lifespan. So by retaining this stemness property in vitro, we realized that we can expand them long term. Mm-hmm. And we totally defied this Hayflick limit, meaning that we can propagate stomach, but now we also can propagate liver and pancreas and lung in the dish for months and months in culture by splitting them once a week, meaning many, many passages. When the limit was 5 passages, now we can get to unlimited passage for the moment. for both mouse and human cells, and still they retain the genetic stability. So this is what we mean that organoid cultures and that are starting from tissue-derived organoids that activate the stem cell program in the dish, or they retain the stem cells that were already in the tissue, they actually in the dish are able to propagate long-term without having this Hayflick limit.
And you also talk about mechanisms of liver cancer and how your systems can be used to study that. So your findings on normal organoids, how can they help with a better understanding of liver cancer?
So one thing that we have realized very early on is that, and it was unexpected because at the beginning we thought that these differentiated liver cells that activate the regenerative program, we just thought that it was just they activate proliferation. But actually what we have seen by studying at the molecular scale is that there is a massive epigenetic reprogramming. So the DNA, the whole genome is kind of changing its epigenome from a quiescent differentiated epigenome state to a proliferative regenerative epigenome state. So what we've seen that, for instance, one mark that is a silencing mark, which is called DNA methylation, that prevents the regenerative program to be on in a quiescent liver cell that doesn't need to proliferate. The upon damage, the massive change on the epigenome makes that this quiescent state becomes activated state because the epigenome becomes permissive to activate the program.
And for the study of liver cancer, you're using human liver cancer patients.
Yes, exactly.
So you can try different drugs.
Yeah, so we can take a cancer from a patient, a tumor, we can dissociate it and put it in the Petri dish, and because we try to recapitulate the environment of that the tumor cells see in vivo in the patient, with the growth factors, with the cocktail that we put in the Petri dish, then these cells actually, in a sense, it's like they think they are inside of the body and they do what they would do in the body, so they keep reproducing. And in that case, because they come from cancer patients, they actually retain the genetic mutations. Right. They also retain the transcriptional program of the tumor. And even what I find very surprising is that they even retain the architecture of the tumor, something that was before impossible to reproduce with cancer cell lines. So this is one of the advantages of these models, that now we have models that recapitulate the architecture and hence the cellular interactions between the cells in the tumor as well.
So in your talk, you mentioned that you develop organoids from healthy and diseased systems, human and mouse, adult and embryonic, and we just discussed why it would be advantageous to study healthy and disease. But why have you also focused on human versus mouse or embryonic versus adult?
Very interesting. So we have learned a lot. For understanding regeneration, there is many principles that have been found from studying animal models and model organisms. However, we still don't know how many of these principles are still conserved in humans and how many of these principles are not conserved, and therefore humans, we have specific mechanisms. And why is this important? Because actually, we are not mice, we are not animal models, and for instance, our immune system is different. We respond differently to toxins, we respond differently to viral infections. And actually, all of them contribute to repair or to the damage and the repair response. So what we have, we always try to use the mouse as our tool set to manipulate, because we cannot go chopping the liver of people, right? But we can now give a toxin to the mice and see what happens to this liver. But we always want to validate what we see in the mouse on the human setting. And of course, because we cannot go to the human patients, we have to have in vitro models that recapitulate human in a dish. And that's why we constantly try to develop our human models further, to have our toolkit where we can play and where we can investigate whether what we have found in the mouse indeed applies to humans or not.
Well, unfortunately, that's all the time we have today. Thank you very much for taking the time.
Thanks for the interview.
And thank you for watching.